Conservation, who will be introducing Tracie for today's talk. Over to you, Rob.
Thank you very much, Lulu. Yeah, thank you all for coming. Great to see so many friends of Saola and the Saola Foundation here. As you know, we are embarking on a really great challenge to find Saola so that the animal can be saved. And we can't do it alone. And for this task, we've assembled a cutting-edge technical program with a number of, well, 3 key partners. And this program is indicating, as you probably know, sign detection dogs with human wildlife trackers and rapid field DNA testing capacity. As I said, we can't do this without these partners. And today we're going to hear from one of them, the WCS Molecular Diagnostics Laboratory in New York. In fact, WCS has quite a long history with Saola conservation in Laos. In fact, myself and our technical director, Rob Timmons, and the father of our Lao programs director, Olay, we were all working for WCS in Laos not long after Saola was discovered there. And in the latest, and we also, in the latest chapter, we have a cooperative program with the WCS Lao program to help us implement the field work. And this key partner in the molecular diagnostics lab of WCS. And in the past several months, the lab completed development of the first ever Saola-specific rapid field DNA test kit. And this is just going to be a revolutionary advancement and aid to our search. As Lulu mentioned, our guest today is Dr. Tracie Seimon. Dr. Seimon is director of the WCS's Molecular Diagnostics Lab. And there, she directs the development and implementation of molecular diagnostic technologies that can be used to search for wildlife and endangered species like Saola through the uses of environmental DNA, and also involvement of things like identifying species in the wildlife trade and diagnosing infectious diseases in wildlife. Dr. Seimon received her PhD in experimental pathology from the University of Colorado Health Sciences Center in 2003. And after that, she served as a postdoctoral research fellow in various capacities between Columbia University in New York and WCS up until 2012. She's the author of more than 60 peer-reviewed scientific publications, and she enjoys communicating her work, as you'll learn today, and also inspiring and mentoring students. In her spare time, such that she may have some of it, she loves to photograph life in all of its forms and natural phenomenon. And for fun, on their vacations, she and her family travel to Tornado Alley in the U.S. Great Plains to study and document severe storms. So she's clearly quite an interesting person, and I'm very much looking forward to her interesting talk. As noted, her talk is free, but if you would like to express your appreciation to Dr. Seimon for a talk, a goodwill donation to the Saola Foundation and our search would, of course, be greatly appreciated. And Lula will put in the chat a link to where you can do that. Finally, on March 25th, about this same time, we will hear from another of our partners. We'll have Lee Gutteridge and Kirsty Lawrence from the Cyber Tracker Network based in South Africa. And they're going to talk to us about the ancient art of wildlife tracking and how they're going to use this tool today to help us find Saola in the 21st century. And with that, I'll turn it over to Dr. Seimon.
All right, thanks, Bill, so much for the introduction. It's an absolute pleasure to be here to talk with you today. Let's see if I can share my screen. Share. Okay. Can you guys see my screen?
Yes.
Great, great. So as Bill mentioned, I'm the director of the Wildlife Conservation Society's Molecular Lab based at the Bronx Zoo. And as you can see from the title of my talk, from Everest to the Annamite Mountains, our lab covers a wide geographic range of various projects throughout the world. And so I'll be talking to you about a subset of some of these projects with applying molecular tools for conservation. And then I'm going to spend the last third of my talk focusing on one of our most exciting projects in collaboration with the Saola Foundation in developing a new molecular tool to better track Saola in the environment. So our lab, our WCS lab, is based at the Bronx Zoo. We are located within the Zoological Health Program of WCS. We are a small lab, though we cover quite a number of different projects. We do pathogen testing for the collection and several of our field-based programs. WCS is in over 60 countries, so we work with our field conservation programs to help also implement tools that can be used in the field by researchers and conservationists. We also have several projects devoted to DNA barcoding for conservation, so I'm going to talk a little bit about that. And then the newest area we've been involved with for the past several years is developing environmental DNA detection, which is DNA that's shed from organisms that live in an environment. And this is a really up and coming, very expanding field that has potential enormous implications, both for in the search of rare and threatened species, as well as biodiversity assessments. So for pathogen testing for our collection, we've been involved with a number of projects. We, of course, do a lot of the pathogen testing for our WCS 5 parks. And in addition to the aquarium, we do pathogen screening for several of our field programs, for example, pathogen screening for bog turtles and other turtles in the Northeast. We work with our collaborators in the Grand Cayman Islands we developed a new test to screen for this pathogenic Helicobacter that was associated with blue iguana mortalities. And then we also do a lot of pre-release pathogen screening for reintroduction programs that include Burmese star tortoises, southern river terrapins, and Burmese roof turtles. And then we extend that to pathogen testing of confiscated turtles from the wildlife trade. And then we've spent a number of years also assessing chytrid risk in various places throughout the world, including Peru, Africa, and Southeast Asia. And then what I'll focus on also is some of the work we've done with developing canine distemper virus diagnostics for our field-based programs. So we do a number of different pathogen screenings, as you can see here from our list of viruses and protozoa and fungi. And then we also focus on lab-to-field technologies. And this becomes really important because, as I mentioned, WCS is in so many countries where diagnostics are critically needed for our health program and our conservation programs. So over the years, we've honed and figured out ways that we can shrink down this molecular technology to be able to take it to the field and be able to set up mobile diagnostics. One of our earlier deployments of our mobile lab was for health assessments of captive reared Burmese star tortoises prior to reintroduction. And so we packed up our equipment in action packers and flew to Myanmar and set up a lab. In this particular picture you see here, this was set up in a hotel lab where we were screening a number of samples that were collected from captive Burmese star tortoises prior to reintroduction. And in some cases, we actually had to take the lab more further into the field to more remote sites, and we set up the lab in a shelter that's right at the captive rearing site near Began. And in this case, we set up our lab in the shelter, which had no external power, so they were able to bring in a generator by oxen cart and help power all of our molecular equipment, like our PCR machines, and we screened, I believe, over 300 samples for a variety of different pathogens of conservation concern that will inform health assessments and pre-release health screening to determine which animals will be released into the wild. So other projects we've been involved with, this photo you see here on the lower left of this tiger, this is Morka. She was a research tiger that was followed by WCS. She was radio collared for over 8 years. And one day in 2010, she abandoned her 3 cubs and wandered into a village of Ternay, and started hunting dogs. And she came too close in the village and was threatening people, and they had to euthanize her. And it turned out that she abandoned her 3 cubs because she had contracted canine distemper virus. And in 2010, canine distemper actually took a pretty big toll on tigers. Her mate, Ivan, went into a village and attacked a fisherman and ended up being shot. And he too was positive for distemper virus. And of course, the 3 cubs that they had, she had abandoned, all died. So when we think about Amur tigers, there are about 500 left in the wild. Distemper is actually posing a significant risk to these populations in the Russia Far East. So to be able to better monitor for distemper virus, we ended up, this is Martin Gilbert shown here on the left. He was doing his PhD studies to better understand how canine distemper virus was spilling over into tiger populations. And so over 2 years, he was trapping mesocarnivores, such as shown here. This is a raccoon dog, and collecting samples from these animals to better understand which animals are carrying distemper and how it's spilling over into tiger populations. And so one of the things that was critical to be able to monitor for distemper virus was to be able to build capacity for canine distemper diagnostics in the Russia Far East. So this is Nadia Sulikan. She was also doing her PhD work on distemper in carnivores in the Russia Far East. And so I traveled over there to help develop capacity for canine distemper virus. And in the several weeks we were there, we screened a number of samples and helped build the capacity within the laboratories in Vladivostok. However, despite the fact that we had now distemper testing up and running in the Russia Far East, it still seemed to be a really big lag between the point of being able to collect those samples and then the testing, which would often take many days to even weeks to get results. And so to help with better diagnostics at a point of care level, we developed a portable canine distemper virus test, which is a qPCR test. And it's in a portable platform, so you can actually take it to the field. And so shown here is the workflow of how this test works, where you collect a sample. This could be from a tiger or other carnivore species, place the sample in a tube with lysis buffer. And the lysis buffer basically opens up the cells, bursts them open, and releases all the DNA into the solution. And then that solution is sucked up by a syringe here, which has a filter that binds to the DNA or the RNA sample, and that you suck it up and run it through there. So all the nucleic acid or the DNA and RNA binds, and then you run it through a series of washes to purify the sample and clean off all the debris and the protein. And so you're left with a pure RNA sample in the green tube here. And then that sample is then transferred to tubes, which then go into the PCR machine. And within 60 minutes, it will tell you if that sample is positive or negative for distemper virus. And so over the next year, we developed this point of care test. And this is Anya Tomasowicz. She helped develop the test over the course of a year, and then she traveled to Russia to train our colleagues so that they could be able to utilize this test at the point of care when they actually have a tiger that's in the field, that's maybe perhaps a dead tiger or a collared tiger, or tiger that they find is sick and in need of veterinary care. They can test it right on the spot. So they don't have to wait weeks or even days to be able to get the result. So this was a really powerful way that we could introduce these diagnostics to be able to inform both health monitoring and conservation research. And then we also focus a lot of work on DNA barcoding for conservation. So DNA barcoding kind of is exactly how it sounds. So DNA is a string of different nucleotides, A, T's, G's, and C's, in different conformations and different, basically every bit of your DNA versus my DNA is different. And so you analyze those differences as a DNA barcode or a read. It's almost like an individual barcode, and that's why it's called DNA barcoding. And so we started developing portable PCR tests that could identify species using DNA barcoding genes, particular genes that could differentiate one species versus another. And then also developing tools that could identify species that could then help identify products, for example, in the wildlife trade, which then, if we know, if we can rapidly identify products in the wildlife trade, that informs both monitoring, conservation monitoring, and law enforcement that then can turn around and help enforce those wildlife trade laws. And you can also use it, for example, health monitoring, detection of rare and threatened species, and then also for diet reconstruction. So one of in the last couple of years, with funding through the US Fish and Wildlife, we developed a rapid DNA test that could identify all big cat species that are found in the wildlife trade. And so this test actually will test for all 6 major big cat species, and it will also be able to differentiate each species based on melt temperatures within one PCR reaction. So you just need one PCR test, and it will tell you whether it's lion, snow leopard, tiger, or cheetah, whichever species that we might be testing. And this is really important from being able to tackle illegal wildlife trade, because bones and other products from tigers and other big cat species are a huge market in the illegal trade. And being able to help with conservation monitoring and law enforcement to be able to identify species that are coming through the illegal trade is a huge help to be able to both understand the scale and the scope of the illegal trade, but also to enforce CITES laws and prosecute those that might be trading in big cat parts illegally. So this test we developed to be able to detect bones and other products such as tiger bone wine and be able to determine what species make up these components. And then that can feed into better comprehensive monitoring and help with law enforcement. So this test has been developed. It's currently being piloted by the US Fish and Wildlife Forensic Lab, as well as our partner lab in China. And then over the next few years, we would like to scale up the use of this test for more comprehensive monitoring of big cat species in the trade. And over time, as we move forward into the future, these tools that we're using are shrinking down and are becoming even more portable. On the upper left is an image of what a DNA sequencing machine looked like when I went to graduate school over 20 years ago. And in the last 20 years, this huge machine that takes up an entire bench top has been shrunken down to a small little box that can literally fit in your palm. On the right is Adeline Xia. She was a postdoc with us for 2 years, and she was helping develop and implement DNA sequencing for identification of species in the trade. And so here she is loading a DNA sample onto the DNA sequencer for analysis. And our other postdoc, Marissa Lim, who's up here on the upper left here, the two of them worked together to both create the protocols as well as the bioinformatic pipeline that can handle all the DNA sequence data. Because when you start sequencing whole samples, you produce a lot of DNA reads, and it's a lot of data that has to be managed by a bioinformatic pipeline. And so she helped develop this pipeline that would lead to the analysis of all that DNA sequence that could then lead into species identification. And then that flow would then feed into enforcement and monitoring. And so that's the idea behind these tools, is that they can lead to faster approaches to where you can actually implement these tools, for example, at country borders and any point of access along the trade route. And so the other area, which I'll focus a little more time discussing, is eDNA for conservation. And this is actually a really exciting field that is expanding rapidly. It's changing rapidly. It's being now applied to a lot of different conservation uses. It can be used for searching for rare and endangered species. It can be used for biodiversity assessments. It can inform environmental impact assessments, and also can be used for pathogen detection. So there's actually quite a number of different ways eDNA is starting to be utilized in conservation. So when we started eDNA analysis back in 2015, we actually started out with a need, much like what I'll talk about later with the Saola Foundation, but a critical need to be able to identify additional individuals of the world's rarest turtle. So this is the Yangtze giant softshell turtle. It is one of the rarest species on the planet. There are currently between 3 and 4 individuals known that exist. One is at the Suzhou Zoo in China, 1, maybe 2, maybe in Dong Mo Lake, Vietnam, and another one in Swan Con Lake. And so without, if additional individuals cannot be found, this species will go extinct. So what was really critical is that there are so many unexplored waterways in Vietnam and China and it takes so long to be able to observe the species. So on the right here is one of the few images that's ever been taken of a wild Yangtze giant softshell turtle, and they surface they don't surface very often, and when they do, it's for not very long. So it actually takes an enormous amount of person hours and survey efforts to be able to observe these animals. So we wanted to know, basically, could we apply eDNA detection, portable eDNA detection, to be able to explore and screen some of these waterways to find additional individuals. But when we started with eDNA, we'd never worked with it before. And we first wanted to find out, does this even work? Because it's such a, concept wise, what we're detecting is eDNA that's shed from organisms in the environment. The equivalent is that you go swimming in a swimming pool, and then I would go and collect water and then test if you'd been swimming in there. So it almost sounded like, when we got involved with it, it was almost like science fiction. So we set out to do a number of different experiments. And the first experiment we did was we collected water from our softshell turtle exhibit on the Bronx Zoo. It's a 950-gallon tank, and it contains Texas spiny softshell turtles. And so we collected samples from the exhibit and then tested to see if we could detect the softshell turtle. And what was amazing is that we detected the turtle in every sample. And we also performed experiments to test half-life of the eDNA, how fast it degraded, but it was a very encouraging start. We then went outside of the herpetology building, and we started testing the water in our turtle pond. And in the water samples, we were able to detect a number of different species like snapping turtle, painted turtles, red ear sliders, and fish species like golden shiner. We also detected birds like common grackle and raccoons, which was quite interesting. So we kept going bigger because, the lake in Vietnam where the one Yangtze giant softshell turtle is known to exist is huge. It's over 4,000 acres. So we had to see, well, can we detect eDNA from larger ponds and lakes? And on the upper left here is Nathan Sweeting, and he had a very large pond on his property with Florida softshell turtles. So he went and collected some samples and sent them to us, and we were able to detect Florida softshells in 5 of the 6 samples on both occasions that he collected for us, which was also very encouraging. And on the lower right here is Paula Dulce Moscolo, who's a volunteer with us, and she took the kit out to collect water samples from Lake Wapelani in New Jersey, which is a 12-acre lake. And from these samples, we were to be able to detect a number of turtle samples. So with that in mind, we started development of the first portable DNA kit that would detect the Yangtze giant softshell turtle. And this kit, the point of this kit was that it could be used on the spot at the side of the lake, where you collect the sample and then immediately test it and get a result. So we developed the test. It took over a year, and we tested both sensitivity and specificity. And we had the kit manufactured and then did the quality control testing. And then when we were happy with how the kits were performing, we then traveled with the kits to Vietnam to train our partners from Asian Turtle Program and WCS in how to conduct the testing. So this image was taken, this is Dong Mo Lake. So it's a huge lake. And during the 2-week training, we collected samples from around the lake, as well as another lake, Swan Khan, to determine if we could detect the Yangtze softshell turtle from 2 lakes which are known to have these individuals. And basically, they were all negative. But over the course of a couple of years, when we determined the sampling strategy and determined the time of year when it was best to detect these, we started getting positive results, which was very exciting. So this is the contraption where we would collect water samples. The water samples would then be filtered through this little filter, which here is Long from our WCS Vietnam program, and he's showing what a filter looks like. That filter then gets extracted, the DNA gets purified, and then it is then tested by PCR, and then it will tell us basically within an hour whether that sample is positive or negative for the Yangtze giant softshell turtle. So we then wanted to see, well, how far can we take eDNA? Can we potentially use it for biodiversity assessment? And in 2019, I was invited by the National Geographic and Rolex Perpetual Planet Extreme Expedition to Mount Everest to lead the biology component of the expedition. And what I had proposed was to basically perform a high altitude biodiversity survey on Earth's highest mountain. And why is this important? Well, up on Everest, it's up there where few life forms are thought to exist. We would be venturing basically up into what we call the Aeolian zone. And the Aeolian zone was a name first proposed by the biologist Lawrence Swan in 1963. And it describes a mountainous thin air environment above and beyond the range of flowering plants. And way up there, few life forms are thought to exist. And that includes plants and mosses that are deposited by spores that get blown up there through the action of wind. And as these plants and other things the pollen start to take up in the soil and develop, the soil starts developing, and then you get little pockets of life developing up in these high alpine Aeolian areas. So what we wanted to determine was what is the highest life and what is the biodiversity above the range of flowering plants. And so this photo is taken up in one of the areas where we were collecting water samples for eDNA analysis. So our goal was to basically determine if eDNA could be used to assess the biodiversity in these high alpine ecosystems. This area where this photo was taken is looking down on the Mount Everest base camp. And you can see there's these little patches of yellow. These are all tents of Mount Everest base camp, all the tents that are basically being pitched on this glacier. This is the Khumbu Glacier coming down the valley. And if you go up this area, this is how you get up through this ramp into Mount Everest. And that's the route the climbers go. So when we arrived, we had expected all of the ponds to be thawed at the time of year we were going. We were actually very surprised to find all the ponds frozen. So that ended up causing us to be able to, we had to hack into the ice in order to reach the water beneath the surface. And so we would create a hole and then insert our filters down beneath the ice and capture the water underneath the ice. And then filter those samples to capture the DNA on those filters and preserve them until we could get them back to the lab. And so after about 4 weeks of collecting samples, we brought our samples back to the lab and processed the data. And this took many months of work and was the result of the hard work of Marissa Lim, who is our bioinformatician. And she basically, her task was to process over 150 gigabytes of DNA sequence data. That's over 800 million DNA sequences that were collected from about 20 liters of water from various ponds and streams above 4,500 meters. And we were quite surprised in analyzing the samples. There was actually remarkable biodiversity up in these remote alpine ecosystems. We detected over 187 taxonomic orders from 36 phylum. Now, when we think about taxonomic orders, 187 orders is about 16% of total known global orders, or about 1/6 of global orders that are known on the planet, all just on this one mountain and above 4,500 meters. And to put that into perspective, the land mass above 4,500 meters only occupies about 3% of global land mass. So remarkable biodiversity up on this mountain. On the lower right here is Tibetan snowcock, which was one of the bird species we detected in our eDNA data. So in addition to collecting water samples, we also collected opportunistically scat samples that were found on the expedition. Several of these scat samples, both from the east and the west side of the Khumbu Glacier, turned out to be from Pallas's cat. And this was a very exciting finding. Pallas's cat has never been before known to exist in eastern Nepal. So not only did our finding expand the range of Pallas's cat to eastern Nepal, but it also confirmed the presence of the species in Sagarmatha National Park, which is one of the most heavily visited World Heritage Sites. So this was the interesting thing, that this species went undetected so long, indicates the power of using molecular tools to be able to search for both rare and elusive species, like for examining scat samples. So in addition to Pallas's cat, we also identified fox scat, as well as prey species that existed in that scat, such as the Royal's pica on the lower right here, as well as mountain weasel, shown here. And the interesting thing is when examining scat, not only can you extract the DNA and determine what's in it, but morphological analysis can tell you a lot of things as well. So this is a picture of one of the scat samples that we had collected, and shown on the right here are tufts of fur, as well as bones that was consistent with the pica DNA that we had identified in the scat. But we also saw was bits of rope, rubber pieces, and plastic, indicating that the trash that's accumulating on Mount Everest is making its way into the food chain of these carnivore species living up there. So it's always important to not only just rely on DNA, but also morphologic analysis and other means to be able to pair with DNA analysis to give you a more comprehensive picture of what might be happening in an ecosystem. So this finally gets me to one of our most exciting projects our lab has worked on with the Saola Foundation, and that is developing a portable DNA test to search for the critically endangered saola. The saola, this image you see here is from a camera trap taken in 2013, and is the last image that's been taken of a saola in the wild, so 10 years ago. And so it's urgently needed that we find more saola in order to prevent them from going extinct through bringing them into a captive breeding program and helping increase these numbers of these animals before they're gone. So we were really excited when the Saola Foundation technical team reached out to us and explained their need. They had been collecting dung in the forest, and the idea was that with collecting the dung, they could use it to better track the animals in the forest. But at the point they were collecting, it took a long time before they could send it to a lab and get that analyzed in far-flung labs, which may take weeks to months to get results. And by that time, that animal's probably moved very, very far away or to other places in the forest. So it's not really an effective way of tracking the animal. And so they reached out to us with a need to be able to develop an on-the-spot test where they could test the dung right away. And that could inform whether the animal had been there recently and allow for better tracking through the use of sniffer dogs. So we partnered with them, and over the course of a year, we both developed and validated the first qPCR test to detect Saola in a forest environment. Now, it was quite a feat because we'd never actually applied our DNA tools, our portable tools, to fecal samples. So we first had to develop a qPCR test to detect Nyala. And the reason why is we had no Saola feces to work with. There is no Saola in the United States captivity. And the only type of sample we had to work with was another bovid species in our collection, which we had access to, called the Nyala. So we first developed a qPCR test to detect Nyala from dung samples to see if the platform could be used successfully to detect DNA from fecal samples. Now, fecal samples are notoriously difficult to work with. They have a lot of things in them that actually block PCR, and that can lead to a lot of false negative results. So it was really important to establish whether this test would work for fecal samples. So we developed the primers and the probes to detect Nyala, and we had the positive control dung material. And then we also screened those primers to make sure there would be no off-target amplification, that would only detect Nyala. We tested the PCR efficiency and sensitivity to get it to be as efficient and sensitive as we could, because that was our goal for the Saola test. And then we basically tested the whole limit of detection of the assay. And what that means is that from the point the Nyala feces are extracted to the point that they're tested, what is the limit of detection? How sensitive is that test? And we need it to be very sensitive. So after we established that this test does indeed work, we went and developed a second qPCR test to detect Saola. And we went through all the validation experiments we did with Nyala, and we basically tested, instead of having Saola feces to work with, we created mock Saola feces using Nyala dung, where we would spike in Saola DNA. Once we were happy with how the kit was performing, we tested it against all these off-target species, so we knew it was going to be very specific and very sensitive. We had the kit manufactured, which took about 8 weeks. And then when we got the kits back, we performed the QC analysis to make sure the kits were performing exactly as we had expected in our validation experiments. So once we had confirmed that, we then moved forward with training our partners at the Saola Foundation in how to conduct the test and analyze and interpret the results. So this is, in a nutshell, what the test looks like. There's not a whole lot of components that are, are huge or cumbersome. There's packets of these, what we call GO strips, which are little tubes that contain all the chemistry that detects Saola in that PCR reaction. And then we have a portable qPCR machine, which is paired to a smartphone. And the smartphone starts the thermocycling, has the software that starts the machine and runs the machine and tells the machine basically how to run the test. And then after the test is complete, 60 minutes later, the data can be uploaded to the cloud. And then anybody who has access to the portal can then log in and look at the results and then provide feedback or data interpretation to the team. So it's a really nice system that's very quick and reliable in getting data both quickly on the spot, and then also it has a mechanism to allow for team members to help interpret and analyze the results. And so down here is the very quick extraction kit. That is basically you move from extraction to PCR, and then data upload are the three main themes. So these images were taken when Rob Timmons visited the Bronx Zoo back in April. On the right is Carol Hanger, who developed the Saola DNA test and did all that work over the course of a year to be able to validate and manufacture the kit. And this is Rob doing a fecal DNA extraction with Carol as part of the training. And then on the right here shows he had just loaded the sample into the machine and is running a test. And then this gives you an idea about the size of the kits. They can basically be placed in an action packer, and those action packers can also be used as tabletops when you're in the field or in the forest to set up the lab on top of the action packers. So after Rob received the training, he then transported various folks from the Saola Foundation transported the kits down to Laos and trained the team there in how to conduct the testing. And so this was amazing for me to see, because we went from training Rob, and now Rob is training the technical team, and now everybody's using the kits, which is really exciting. And I was very excited when I logged on to the portal and saw their very first test that had been done on Saola dung that was archived. It was pulled out of a freezer, and this dung is from 2010. And what you can see here, this is actual data from the run. And the green here, so this green is positive control, and these three are different aliquots of the Saola dung that they tested, and they were all positive. So this was the first time we could actually test the kit on actual Saola dung, and it showed that these kits actually work, which was very exciting. So why is Saola dung so important to identify? This is a species we know almost nothing about. This test could confirm Saola still exists in the Annamite Mountains, because as I mentioned, these animals have not been photographed since 2013. And we can learn more about Saola in one day of testing than we have in the past 30 years using portable PCR technology. It can, for example, tell us the presence and absence of the dung that's found in the forest, and that can inform biogeography and better tracking of the animals. And the dung itself, if we identify positive dung, this is very valuable from providing additional information. So if you recall what I said about looking morphologically at what might be in the dung, and looking for example, what that animal might be consuming, it can be providing information on, for example, compared to other dung samples, to identify individuals within a population. And if you build that database further, it sort of becomes like an ancestry.com for Saola. The dung can be used to train sniffer dogs, and that will allow them to better track them into the forest and monitor them. And then it can also be used for testing for pathogens of conservation concern that might impact the health of the Saola. And then, of course, to assess the natural microbiome using metagenomics and this can tell you baseline information about the microbes that might be important from an animal health perspective. So there's a number of things that and information that we can get if we're able to positively identify Saola dung. So in short, we feel that this could be a game changer in detecting and tracking this rare and elusive species. So with that, I'd just like to acknowledge the Saola Foundation and the Critical Ecosystems Partnership Fund who funded this work, and the various donors that have funded previous projects in the lab, our WCS and molecular program, and various folks who have been on our team over the past decade, as well as the various trainees. So with that, I'd be happy to take any questions. Thank you.
Well, thank you, Tracie. That was absolutely fascinating. And I just want to make a couple of comments before we turn it over to questions. That was fascinating. Thank you so much. And as I hope all of you can tell, we have some great partners in the Saola Foundation, which one is Tracie in the WCS Molecular Lab. And because of these great partners, we've made fantastic progress. And we start the year of 2023 with great optimism and great progress behind us. Just by brief update, in the last few months, both the professional detection dog trainers and the human wildlife trackers have been in the forest with our team doing the first initial assessments. We've completed recruitment of 15 Lao national team members for the dog handlers and the wildlife trackers and the technical teams, recruited from villages in the Annamites and throughout Lao society. And we have just a fantastic group of people. And in fact, in just a couple of weeks, a young Lao woman, Vatsana, who we hired to be the leader of the detection dog handlers, we're flying her to the US, to Montana, to start her training with the detection dogs. So 2023 is the year when, after all these years of preparation and the great input from our partners, we are going to start the search for Saola. And we start it with great confidence, in part due to partners like Tracie. So this is a critical moment, and your continued partnership and support in 2023 is really, really important. And we have such gratitude for all of you. So thank you, Tracie. That was fantastic. One other thing I'll just add, benefit of collecting Saola dung. We have partners who believe they can extract plant DNA from Saola dung to help us get a better idea of a library of plants that Saola eats in the wild, which will help maintain them eventually in captivity. So thank you very much. And I'm going to turn it over to Lulu, who's going to monitor the Q&A.
Thanks, Rob. Thank you, Tracie. That was really fascinating. So just to let everyone know, if we do go slightly over the hour, you can leave. And if there are any unanswered questions at the end, they will be answered in an email afterwards. But over to the chat. Our first question is from Lorraine. She says, she asks, the multiple cat species DNA test for monitoring wildlife trade is amazing. Is there any scope for identifying wild / versus captive born animals or captive products in trade?
That's a great question. I know there's been a lot of interest in being able to distinguish between captive and wild. The kit that we have developed, it's mainly relying on a barcoding gene. So it's not going to be able to distinguish between one that's captive or wild. It just basically will tell you if it's tiger, lion, which big cat species it is. There is potential for being able to, for example, if you can identify particular SNPs that might be associated with captive versus wild, you could potentially create primers that would target something like that. But I don't know that that's been applied using our portable tool yet. I haven't seen that yet, but never say never in science.
That's great. And then we have a question from Marcus. He asks, so a high number of different DNA samples of species on high altitudes, but does it really mean such species inhabit the high mountain area? You noted the wind blows plant and species residues to these parts of Mount Everest.
Mm-hmm. That's another great question. So eDNA analysis always has to be interpreted with caution. So for example, eDNA, you might be also detecting ancient DNA. For example, if you take a lake core and you extract DNA, you might be able to detect eDNA that has been there or preserved in ice cores for a very long time and may not exist in contemporary settings. Also, eDNA has a half-life. So interpreting eDNA results of not finding something does not necessarily mean something's not there. It just was below the level of detection of our assay. As far as like stuff being blown up and windblown, this is a really good point because we found, for example, some of our pollens were pine trees that are likely pollens that were carried up, through the action of wind. But we also found a lot of plant species like juniper, which are only existing, between, that were around in some of the areas where we had detected them. So for example, in some of the streams where we had collected, were surrounded by juniper, and that was the predominant pollen we detected there. So I think you have to be very careful in how you determine it. basically you find DNA there, but it may not completely mean that species is there. For example, also birds could be flying over and maybe not stopping there, but might be dropping fecal samples that you collect and you find in your sample. So these are all things you have to use caution with interpreting your eDNA results.
Yeah, that makes sense. Bill asks, can eDNA testing provide an estimate of population size? Does a positive eDNA test tell only that there is a turtle in the lake or many turtles?
So that's always a question of whether what you're targeting with eDNA testing. So if there's enough research done to where you can find a target that would tell you individuals that you could amplify and then, for example, detect through sequencing or something like that, you could potentially get to individuals within a population. It's just you need the work being done beforehand to be able to identify that area that you would be targeting.
That's brilliant. We also have another question from Bill. He says, I think in reply to CDV, is veterinary intervention possible for cats that test positive in the wild?
So with CDV, the primary veterinary intervention is supportive care if they survive it. So far, I don't know any examples where cats have survived distemper in those settings in the Russia Far East. So supportive care is very important. I think it's more for perspective in informing the veterinarians that are on site that, or if there's a dead animal, whether that animal is positive or not. And so it's a lot more information than just informing the veterinary care, but it might inform, for example, management practices. It might inform, you know, what, for example, an outbreak, what might be going on if they find several animals. So yeah, it's really kind of dependent on the situation.
Yeah. I believe it's Joaquin has asked, can the eDNA test kits be used to detect other Annamite species too? Has this been done yet?
I believe there, and then I would actually, I would punt this to Rob because Rob, I believe, has done some testing in the past with other species. So I'm actually gonna push that. Is Rob here or?
He's not here, Tracie.
Oh,
so
okay.
I'm gonna take a stab on his behalf and say no, I don't believe so. Certainly not that I'm aware of. And Bill, please chime in if you know different. But I don't believe that. But certainly highly desirable.
I've heard there were.
There were some detections of some interesting things when we first tried the leeches. You know, a couple of years ago, we were collecting leeches to try to find Saola DNA. And there were some interesting records, like range extensions that came out from the leeches. But I think to get to Jos's question, just this DNA test kit, it's just yes, it's Saola or no, it's not Saola. If it's not Saola, it doesn't tell us what it is. It's like a COVID test. You either got COVID or you don't. If you don't have COVID, it doesn't tell you what you do have. It's just COVID, yes or no. And this is just Saola, yes or no. So.
That's great to know. And Paul asks, can the test detect male and female individuals?
No, this test cannot. It's really yes or no Saola, but it won't detect gender.
Thank you. That is all the questions we have in the chat for now. We will soon press stop on record and unmute. So if anyone does have any questions and doesn't want to be recorded. Oh wait, we might have one more question. Some phone says, how is the cost for testing sorry, I can't see. Oh, how's the cost for testing a set of sample dung or scats. So how much does that cost?
What is the cost of the test?
Yeah, to test a set of samples, such as dung or scats.
Okay, so I believe I mean, it's hard to say because we purchased everything sort of together for a set of kits. But I think it runs, I think between probably 40 or 50 a sample to test.
Yeah, which is extraordinary, because in the past we would have to collect samples and then get export permits and send it out to a lab, and it cost hundreds and hundreds of dollars and lots of time, as Tracie said. So this is cheap for what we can learn. So it's fantastic.
Yeah, it's brilliant. And Bill has asked another question. So further on CDV, my concern is that this could be a serious threat to Amur leopards.
So we actually had our first case of CDV in Amur leopard in 2015, and that animal did die. So that was one of the animals that it utilized our test for diagnosis. So that made it, in that sense, we were able to learn very quickly what it was ailing from. And but yeah, it is concern. And importantly, it's like, with the work Martin Gilbert is doing and is still doing. Both, I think he just published a paper of CDV in tigers in Nepal, is try to better understand the ecology of how it's spilling over from wildlife itself, from mesocarnivores, what the influence of dogs is at the dog wildlife interface as well, because distemper is also, it's a disease of dogs, and in a lot of these places there's very low vaccine for the dog population. So it's something that is important to both monitor for and be able to respond to quickly in those cases.
Yeah. So before we go to an unrecorded chat, I want to say thank you for answering all the questions. Thank you for the wonderful talk, Tracie. And we're going to go ahead and stop the recording.